Hello everyone, this is the third lecture for the BTY719 Biofuels course, and today we're going to look at selected biotechnology advances to improve industrial biofuel production. So we're going to be looking specifically at some of the scientific research that goes into improving the production of biofuels. So for biofuels to play an important role in meeting our future energy demands, a multidisciplinary approach is needed. So that means that biologists, but also agroeconomists, engineers, energy experts, and policy specialists all of those roles must be integrated. And while we will concentrate for this course and for this talk on the role of biological research, we must keep in mind that the entire value chain requires improvement, and that means all of these other role players are important as well. We need to develop and improve high yielding energy crops. So it starts with our feedstock. We need to improve that. We need to improve the efficiency and sustainability of biorefineries. There are some biorefineries that are in operation today, but for a lot of these, their profits are razor thin, which means that they are likely to be unsustainable if there are big fluctuations in fuel or product price. We need enhanced infrastructure, which means we need to look at the transport and the distribution networks and overall production chains of the products that we're making as biofuels. And we need commercialization and policy support, which isn't even scientific challenges, but are equally important in making biofuels integrated into our society. So we need to be aware also of socioeconomic concerns such as land management practices and choice of biomass source. And this should be carefully addressed so that biofuel production does not negatively impact our food security and biodiversity, as we've mentioned previously. We also have to continue to study the actual environmental impacts of specific biofuels. People who do biofuel research are fond of talking about the potentially zero carbon emissions of biofuels, with the idea being that carbon is sequestered from the atmosphere during photosynthesis to produce plant biomass, and we use that plant biomass to make fuel so that when it burns, that carbon dioxide is returned to the atmosphere. But generally, in terms of producing a biofuel, producing that feedstock, transporting the actual product that you're producing, there is still a lot of emissions from the vehicles that you're using to do this transport and to do your farming. And there is, in other words, a lot of unaccounted combustion that takes place, as well as impacts on water resources and impacts on the soil nutritional concerns. So we must be able to do full life cycle analysis so that we can get a true picture of combustion emissions, which will vary according to the different biofuel and the different feedstock. We will have to look at the complete energy balance that is achieved during this entire life cycle of producing a biofuel, which means that, as I've mentioned earlier, we want to see that all of the energy input into producing and distributing a biofuel isn't actually more energy than what is inherent in the biofuel itself. And in the same breath, we have to look at the environmental footprint of the fertilizers and the herbicides that is used during production of the feedstock for our biofuel. So the picture is significantly more complicated than just talking about a closed carbon loop. So we're going to spend a lot of time in this lecture on lignocellulose as feedstock for second generation biofuels, and I'll just remind you quickly of the three main components of lignocellulose. We said that cellulose, hemicellulose, and lignin make up this part of the secondary cell walls of plant cells, which gives them the structural strength. And two of those polymers, cellulose and hemicellulose, are sugar polymers, which can potentially be used for the production of a biofuel. Cellulose are homopolymers of glucose that are linked with beta-1,4 glycosidic linkages. There's a lot of hydrogen bonds between these long continuous glucose chains, which allows them to pack very closely together in microfibrils and fibrils that exclude water, and we call that a crystalline structure. Hemicellulose consists of a variety of sugars, among which xylose is normally the main component, depending on the plant that you are talking about. Arabinose is the other big 5-carbon sugar along with xylose. You also get glucose, galactose, and mannose as sugars in hemicellulose. It's a far more branched and amorphous structure, which means it's more open and can allow enzymes to get in there a little bit easier, which is also why hemicellulose breaks down faster than cellulose during pre-treatment processes. Forming a matrix with cellulose and hemicellulose is lignin that is actually covalently bonded to hemicellulose. These are phenylpropane units, which means they're phenolic units, they're not sugar units, and the breakdown products cannot be fermented. They are highly polymerized structures that play kind of a glue-like or a cement-like role in bonding the fibers of cellulose and hemicellulose and give plant cells that rigidity and that strength that it needs. So lignocellulosic biofuels, we can always think of it as a complete value chain, which starts with primary biomass production. Our feedstock needs to be produced somewhere. It doesn't matter what type of feedstock it is, if it's a energy crop that was grown specifically for biomass or for biofuels production, or if it's an agricultural waste. That then has to be transported to a processing facility which might be a biorefinery where we're going to make our product of interest. We are going to require primary biomass processing, which is often size reduction, etc. We'll talk a little bit more details just on the next slide. Then we have the various aspects of biomass conversion to biofuels and byproducts, which are including pretreatment and the hydrolysis with enzymes and the fermentation, and finally distilling our ethanol or whatever biofuel or product we produce. And then finally there's the marketing and distribution. We have to distribute our product to where it is needed, and it has to be taken up into the society. And we see that when we consider biofuels, or advanced biofuels at least, it's important to consider this entire value chain. As I mentioned earlier, we will tend to look at a lot of research around this section, but it is important to keep everything in mind, as all of these are different steps that we require on this ladder for successful implementation of biofuels. So while improvements are required across the entire value chain, not all of these are biotechnological or even scientific challenges. Product distribution and marketing is not a scientific challenge, but certainly is as important as anything else in this value chain. So a multidisciplinary approach with cooperation of all of these stakeholders involved at these various processes is absolutely vital for the success of advanced biofuels. So let's look at some of the research that is envisaged to improve the entire value chain. As I said, we have research and development across the entire value chain, and we will take them as different sections here. So in terms of primary biomass production, the choice of biomass is very important. Are we using specific energy crops, something like those giant grasses like miscanthus? Or are we looking at traditional crops? So are we looking at agricultural waste, wheat straw, or sugarcane bagasse, or corn stover, all of those non-edible parts of the plants that are left after we've taken away the main product. It's very interesting, this type of research. For example, with sugarcane, there are breeds of the sugarcane plant that grow to a massive size, but they yield the same amount of sugar per hectare as some of the other strains. But in a field where we might be interested in the sugar and in a large amount of biomass, those types of sugarcane strains become very interesting. We'll discuss some genetically modified organisms that are being developed to increase the growth of plants on marginal land. We'll look at some GMOs as well for improved biomass hydrolysis. In other words, plants that are literally easier to break down to release the sugars and help us with this process of biomass to biofuel production. And we also require good production policies here to regulate food security. As I said, we don't want to use all of the biomass that we produce as food and turn that into fuel, and we also don't want to use all of the land that is required for our food security and turn that over to biofuel production. Biomass transport, with respect to feedstock and product value and energy balance, how far can feedstock be transported? It's generally thought that that's because of the energy density of most of the types of biomass that are used for biofuel production, you can't transport it more than about 30 kilometers, 30-kilometer radius from your biorefinery. So your biorefinery location becomes very important because you have to have a link between your feedstock production, your biorefinery, and then the market where you are going to sell your products. You can't be transporting these things too far. Another consideration is a year-long feedstock supply. That is very important for a lot of the agricultural waste that is considered as feedstocks for a refinery. They're only available for a few months of the year. So do you bring in enough feedstock so that you can store it? How is it stored? How long can it be stored? Those are all important considerations. So in terms of primary processing of the biomass, this entails size reduction options. We said that we need to cut and mill our biomass to a very small size to make the downstream processes more effective. There are interests in energy densification, which might mean that we spoke about this biomass transport just earlier. Would it make more sense to transport biomass further if we have central points between farms, let's say, where we have some kind of energy densification process, some kind of pelleting of the biomass? Torrefaction is one option that is being used, which is pelleting and heating. Does that make sense? That's an interesting engineering challenge. We have to talk about feedstock blending and homogeneity as well. All of the different biomasses that we consider for biofuels, we mentioned that they have differences in terms of the amount of cellulose and hemicellulose, and you can't just develop a process for corn stover and think it's going to work equally well for any other kind of biomass that you might bring in wheat straw, for example, because the composition of wheat straw is completely different. So do we bring these different types of feedstocks together and mix them before they go into our process so that our process always basically sees the same mix and we optimize everything for that? There's significant research going on for that as well. And then feedstock storage, as I mentioned, how long can you store a feedstock? It's a biologically active material, so it changes over time. And how does that affect our production of our biofuel? Then what we'll probably spend most time on in this course falls in biomass conversion to biofuels and bioproducts. We'll look at different pre-treatment options. We said we need that pre-treatment of our biomass to open up the fibers of cellulose and hemicellulose in order to get our enzymes to those polymers. And the next one is enzyme cocktail optimization. We mentioned the different types of enzymes, the cellulases, the hemicellulases, that are required for complete breakdown of the sugar polymers to release the sugars that can be fermented. Biomass fermentability is an interesting area of research. I did also mention that all pre-treatments yield some sort of toxic inhibitors in your fermentation broth as well. Hydroxymethyl furfural, phenolics, the first two being sugar breakdown products. And that has a big impact on your fermentation organism. So there's a lot of research that's going on there as well. Which process configuration options are best, and we'll discuss at the end of this first part of this third lecture. I'm going to spend some time on different process configuration options, and we will see that there are advantages and disadvantages to the four main types that we'll discuss. Then, in terms of genetically modified organisms, again, pentose sugar utilization is very important. Pentose sugars from the hemicellulose are a big part of the overall percentage of sugars in biomass, and we need to be able to effectively ferment both the hexoses and the pentoses in order to make the overall process economically feasible. Now, most good fermentation organisms, the ones that are most used in industry, yeast Saccharomyces cerevisiae, which is by far the most used, but also some of the different types of bacteria that are used, they generally don't ferment pentose sugars as well. So they don't ferment those 5-carbon sugars like xylose very well. And even those that are engineered to do so, or some bacteria that do so naturally, don't do the fermentation of glucose and pentose sugar simultaneously. So there's a lot of research that goes on in that regard. Then we'll spend some time on consolidated bioprocessing, which is a conversion option where we get a one-step conversion of pre-treated biomass to our product of interest. And we will discuss some development of these types of organisms that are required for consolidated bioprocessing. And also alternative products. We will generally spend a lot of time, and a lot of the papers that I'll give you to read in the course will be about ethanol production, but there are other alternative products that might be higher value. We mentioned butanol in the previous lecture as well. So there are a significant amount of research that goes into making these alternative products, or we might want to make alternative products for a biorefinery. And then finally, in terms of the product and marketing or product distribution and marketing, we have to keep up with market demands. We cannot oversupply the market because that leaves our price of our products will go down significantly and might make the overall process no longer economically feasible. We have to look at distribution options and requirements, and we have to look at the overall impact of the distribution on our overall energy balance. So you'll see it's a large amount of research from different fields and different disciplines that are required to optimize the conversion of lignocellulosic biomass to a fuel or product of interest. Okay, so we're going to start off by looking at some of these things that I mentioned in terms of how to improve the process. So we'll look mainly at the biological challenges. And the first is the feedstock improvement through plant biotechnology. So the US Department of Agriculture has estimated that about 1 billion dry tons of biomass per year would be needed to replace 30% of the transportation fuels that the United States will require by 2050. And with feasible technological advances, this amount could be produced by 2050 from crop residues, so agricultural waste, as well as some energy crops where they will grow trees or giant reeds or miscanthus, and they will still be able to meet the food, fiber, and export demands. So they can produce enough biomass to replace 30% of the fuels that they will be requiring at that stage. But the biotechnology challenge is that we have to substantially increase crop yields and develop crops with a suitable chemical and physical trait for energy production. So plant breeding and biotechnology can be used to improve our energy crop yield, and this is the simple mass per hectare yield of those energy crops, to improve the tolerance of those crops to pests and drought. Very important, because this might allow us to use land that is not suitable for the production of crops for food. We might want to alter the characteristics of the plant through these breeding and biotechnology methods. For example, there is some research that shows that getting a lower percentage of lignin would be better for pre-treatment processes, getting an increased oil content in terms of producing biodiesel. That would be very interesting, and it could make it more efficient to convert our plant biomass to liquid biofuels. And then also we'll look at modifying plants to produce specific chemicals or even to express enzymes that facilitate processing. And we'll look at some advantages of these. First, to increase the plant yield. So plant growth can be improved by increasing the efficiency of light capture during photosynthesis and introducing genes from photosynthetic bacteria has been shown to allow this to happen without affecting changes in the activity of plant-specific genes. Any of these GMOs that we make, we don't want to lose the aspect of the organism that made us interested in the organism in the first place. For example, if we want to genetically alter a maize plant in order to make more biomass, that would be great, but we can't do that at the expense of the maize production, of the actual grain production. That has to either also improve or at least remain the same. So it was shown that manipulation of genes involved in the metabolism of nitrogen, so overexpression of glutamine synthesis gene in poplar trees increased the tree height quite significantly. So that is one way of increasing the plant yield per hectare. There's also research into extending the growth phase of plants by reducing seed dormancy or by preventing or delaying flowering to allow the plants a longer time to grow. There is research into raising plant protection to abiotic and biotic stress. Abiotic stress is the primary cause of crop loss worldwide, and it reduces average yields by over 50% with additional losses due to pests and pathogens. And in the biotechnology department at UWC, you will be aware that there's a large number of plant biotech groups, and then the research into plants that are more or that do better against abiotic stresses is an important aspect of that research, and this is all to do with improved food security. So developing crops with improved resistance to stress and equipping plants with enhanced resistance to pests and pathogens are at the center of numerous of these types of crop improvement initiatives. So transgenic rice that overexpressed the chloroplastic glutamine synthase was shown to increase the tolerance of the rice with improve the tolerance of rice to soil salinity. So there's also research into optimizing the chemical and physical attributes of these feedstocks, development of energy crops that are specifically tailored with desired chemical and physical characteristics. So attention here shifts from the plant grains to the plant stovers. The stovers is the dried leaves and the stems that remain, that in maize production, for example, we're not interested in. And also towards trees and perennial grasses and low-cost agricultural and municipal waste. So synthesis of cellulose and lignin in plants are co-regulated, which makes sense. As I said, they form this interlinked matrix, so their synthesis is going to be interrelated. And researchers have looked at reducing the proportion of lignin in a plant so that it can increase the proportion of cellulose. So cellulose essentially makes up the weight that is lost during lignin, and the idea is that you would then increase the cellulose percentage in the plant and decrease the lignin percentage, and hopefully get the same amount of plant strength. And that would facilitate easier access for hydrolyzing enzymes because there's less lignin, and facilitate easier pre-treatment. There's all kinds of possible advantages there. So poplar trees with the cinnamomyl coenzyme A reductase, where that enzyme was suppressed, that reduced the synthesis of lignin. Novel strains of switchgrass was produced with lower amounts of lignin, and plants were also produced with decreased or altered amounts of hemicellulose. And this might also be interesting because we can theoretically then decrease the 5-carbon sugars, the C5 sugars or pentose sugars, and remove those side chains to make the process of turning your biomass into fermentable sugars a little bit easier pre-treatment methods and easier enzyme hydrolysis methods. The problem with all of these are that they often produce plants that are not as strong as the originals, which means that you end up making a plant with a better chemical composition in terms of what you might want for biofuels production, but you are producing a plant that cannot grow as well, and therefore you're not producing enough or the same amount of plants per hectare of land. But there's still a lot of research in this, and in some of the Journal Club articles that we will see in this course, I have paid attention to that as well. So some interesting work is from a company in America called Agrivida. They develop seeds that are engineered with pre-treatment and cellulose degrading traits. Specifically, they've developed plants that produce in planter, as the plant grows, some of these cell wall hydrolyzing enzymes. I've specifically seen a presentation at a conference where they spoke about expressing bacterial endoglucanases. Now, the one problem with that, of course, is if you are expecting a plant to grow and at the same time you're making an enzyme that breaks down cellulose, you might expect that to be a problem. So you have to control the expression or the activity of that cellulose that you're producing in the plant. And they do this either through heat activation, where the endoglucanase that they express is actually only active under very high temperature, or under higher than ambient temperature at least, so that it's not active under the normal growing conditions of the plant, or something called intine splicing. Now, if you're not familiar with that, an intine is a segment of a protein that is able to excise itself and join the remaining portions, called the extines, with a peptide bond in a process termed protein splicing. And it's similar to almost thinking of protein introns and exons that splice together to form an mRNA that allows production of a certain protein. Here it is just a section of protein that is spliced out in response to a certain signal that then allows an active protein to be formed. So if you're producing your endoglucanase in the form of an intine, or that contains an intine, it's not going to be active before a specific signal is given and that intine is spliced out. So we can control the activity so that it's only active when it needs to be. A big problem with a lot of these technologies is that again, they are producing, they work theoretically quite well in the lab setup, but in field trials, they often find that these plants are not as strong as they need to be and do not leave the same or give the same amount of production level as the parental strains. So we're going to talk about different biomass conversion options next. So just to remind you again, we start with our lignocellulose feedstock. We spoke about biological and thermochemical processing methods. The biological route that we'll spend most time on requires pre-treatment of your feedstock, then enzymatic hydrolysis, then fermentation of the different types of sugars to ethanol, and it is these steps that are combined in consolidated bioprocessing that we'll also spend a lot of time on in this lecture. Your ethanol is then distilled and you get your cellulosic ethanol, which you can sell in the market, but you will also get some residues for thermochemical conversion or producing a biogas. The thermochemical routes include simple combustion in a boiler, just simply boiling your lignocellulosic feedstock and producing steam, which can be used to make electricity and heat, which may be required for other processes. We mentioned pyrolysis, which is heating in the absence of oxygen. This produces charcoal and pyrolysis oils that can be upgraded and refined for a transportation fuel, although this is a fairly messy oil and it takes a lot of upgrading, which is quite expensive. The other option is gasification and syngas production, which can be used for heat and electricity production in itself, or through synthesis process like the Fischer-Tropsch processes and refining, you can produce a finished transportation fuel. Biogas can also be produced from these residues that we got from distillation, and it can be used as a cooking fuel or to produce electricity in the process. So the conversion of biomass to ethanol, as you can see, is quite a complex process, and we need advances at several stages for efficiency and cost effectiveness. Four biologically mediated processes are involved in the biological conversion. So this conversion starts with pre-treated biomass and ends with ethanol. So to get us from pre-treated biomass to ethanol, we require enzymes. So those enzymes are normally produced separately. We then put those enzymes onto the feedstock and hydrolyze them, start releasing our sugars. We then ferment those sugars, and this is normally also a two-step process because the organisms that are very good at hexose fermentation are not good at pentose fermentation. So separate vessels for hexose fermentation and for our pentose fermentation. Okay, so there's been quite a lot of research and a kind of an evolution of this biomass processing configurations over time. And we recognize four processes or four strategies of how to convert biomass to a biofuel. The first and in many ways simplest is the separate hydrolysis and fermentation, SHF. And we have separate and perhaps even off-site enzyme production where we require probably filamentous fungi to make these large concentrations of enzymes that are required in the process. We then take our pre-treated biomass and we add that enzymes to that, and we get our substrate hydrolysis. And then, as I mentioned, we have hexose fermentation and pentose fermentation likely taking place in two different vessels because we need two different organisms or consortiums of organisms to do those two differently optimized processes. So what you see here, it's four separate vessels that are required to do the four biologically mediated processes. Any kind of improvements of these processes are normally done with consolidation or combining of two or more of these steps. First one, which is actually also commercially used, SHF is actually a commercial process for cellulosic ethanol production, as is SSF, although not as widely. You still have your separate enzyme production, but here they are combining the substrate hydrolysis and hexose fermentation steps. So in other words, instead of just adding your enzyme to the process, allowing the hydrolysis to happen, and then taking the sugar that is produced here and allowing or adding your yeast, for example, and allowing hexose fermentation, in SSF, you will basically add your enzyme and your fermenting organism at the same time. So you have simultaneous saccharification and fermentation. As the enzymes sacharify your substrate, your fermentation organism is already starting its fermentation process. And we'll see that there are some advantages to this first process on the next slide. The third option would be something called simultaneous saccharification and co-fermentation. So here we've combined the hydrolysis and both of the fermentation steps. So you have an organism or a consortium of organisms that can use both hexose and pentose, and it's similar to SSF, only you're using all of the sugars. And then the final step, and the one that we'll spend some time on in the second part of this lecture, is so-called consolidated bioprocessing. Now, the thing about CBP is you will need an organism or a consortium of organisms that produces its own cellulolytic enzymes that'll allow substrate hydrolysis, and then that same organism or consortium will ferment both the hexose and the pentose sugars. Okay, so quite a lot of information on this slide, but it's important to talk through it because we are comparing these different process configurations, and we are going to point out that they all have some advantages but also some disadvantages. So the first one, the one with the four different vessels, that is separate hydrolysis and fermentation. It's a mature technology. It's already being applied in industry. So it's a current industrial process, and that means a lot of work has already been done. A lot of optimization has already been done. We know a lot about this process as well. It also means that there are optimized enzyme cocktails. Companies have made enzyme cocktails that are tailored specifically for this process. There are also optimized pre-treatment and fermentation technologies that basically fit in this entire process across different parts of the value chain. And so the different aspects are basically being optimized to work together. An important aspect here is that we can also have optimal temperature for hydrolysis and fermentation. So what do we mean with this? If I go back, here we have, in this SHF process, we see that our substrate hydrolysis and our fermentation steps are separate. And it's important aspect this because the enzymes will tend to work better at a higher temperature at around 50 degrees Celsius. Even mesophilic enzymes from mesophilic organisms tend to work better at 50 degrees Celsius. It's simple enzyme kinetics. You can't push the temperature up too high because then you get simple denaturation of your enzymes, of course. But the point is 50 degrees is round about an optimal temperature to do substrate hydrolysis at. The fermentation organisms that we use, be it yeast, Saccharomyces cerevisiae, or Zymomonas mobilis, a very efficient hexose fermenting organism, they actually grow at much lower temperature. And so in this process, you can, because you have them in different vessels, you can have this process at 50 degrees Celsius and this process at 30 degrees Celsius, and both your hydrolysis and your fermentation happen at optimal temperatures. However, there are several disadvantages to this process as well. You've got a large amount of enzymes that you need. You've got 4 separate vessels, and both of those mean that it's a big increase in cost. It's an expensive process. You don't get co-utilization of your sugars in this process, which takes the productivity and the length of operation to much longer times. You get feedback inhibition by the sugars. So as you are releasing, for example, more glucose during the hydrolysis step, the glucose will start inhibiting beta-glucosidase, and you'll get salibios that will start to accumulate, and that'll inhibit your salibiohydrolases and your endoglucanases. Because of all of those factors, you get relatively low productivity throughout the process, so a low gram per liter per hour ethanol production. And because you're making a sugar syrup before you're fermenting it, you get a higher risk of contamination. Especially in industry, these things don't go into an autoclave, they don't get sterilized. Other bacteria might get into the process as well, and because you're producing the sugar syrup in your hydrolysis step, you might get bacterial growth. In the simultaneous saccharification and fermentation, where we combine those substrate hydrolysis and hexose fermentation steps, we get a removal of that feedback inhibition, and we also decrease the risk of contamination. And both of those are because the fermentation, as the fermentation happens, it keeps on removing the free sugar. So as the enzymes hydrolyze the substrate and some free sugar is released, your fermentation organism takes up that sugar and uses that for the fermentation. So we get less feedback inhibition and less contamination. It's also a relatively mature technology. It's also being applied in a few industrial scale fermentations. You get improved productivity rates in terms of your gram per liter per hour, so you get a shorter fermentation time overall. You still require a large amount of enzymes to make SSF work though. You still require 3 separate vessels, so those are big cost factors. You still don't have co-utilization of sugars, your hexose and pentose sugars. You still need separate fermentations for those. And now you run into this problem of non-optimal temperature for hydrolysis and fermentation. You're picking a compromised temperature where your enzymes might still work pretty good, but your fermentation organism will still ferment. It's not an optimal temperature for either of those processes though. Then for simultaneous saccharification and co-fermentation, again, you're removing your feedback inhibition. Your pentoses and hexoses now get fermented in one reactor by one organism or a consortium of organisms, and you get improved ethanol yield, which is critical to make our downstream processes a little bit cheaper. The higher the ethanol production that we can get in this step, the cheaper the distillation process becomes and the cheaper our biofuel becomes overall. However, it's an immature technology. It's not being used in industry. It's still really in the research phase. You still need a relatively large amount of enzymes to be added to the process to get complete breakdown of your feedstock. You still have the problem of non-optimal temperatures for hydrolysis and fermentation, and you still need to optimize your organisms for this co-utilization of your hexose and pentose sugars. As I said, not only are good glucose fermentation organisms, the wild types don't use xylose, for example, at all. Even in organisms that use both of the sugars because of catabolite repression, you get utilization of the hexose sugars first, the glucose specifically first, before you get utilization of anything else. Then finally, the overall dream would be to produce a consolidated bioprocess where you have one-step conversion of pre-treated biomass into ethanol. And this would remove feedback inhibition. You would have no exogenous enzymes required in a complete consolidated process, although you might add some enzymes to get the process started, but this would then theoretically be far less enzymes that are required. And economically, in economic modeling, this is supposedly the most advantageous of the different type of configurations. However, there are no optimized organisms that are available to do consolidated bioprocessing. We are trying to engineer some. So it is also an immature technology. You again have the non-optimal temperatures for your hydrolysis and fermentation. And the organisms that we do develop might also turn out to be very feedstock specific because of the differences in the amounts of cellulose and hemicellulose in our feedstocks. We might optimize a CVP organism for one feedstock and one type of pretreatment, but a different one might have to be made for a different feedstock and pretreatment. And there might also be intellectual property hurdles. Everybody who develops organisms for consolidated bioprocessing is normally very quick to publish a patent, and one patent may interfere with another researcher's ability to make an optimized CBP organism. So all of those are problems that still need to be overcome before CBP can become an industrial reality. Okay, I'm going to stop this part of the lecture here, and we will continue in the second part of the lecture with development of CBP organisms. We will look at some metabolic engineering as well as the biorefinery concept.